All right, here we are, and I'm your roving biologist. And lab two, I want us to look at some groups of protists that are symbiotic, in particular those that are parasitic. And because we're all scattered, it's going to be hard for everybody to do exactly the same thing. And I'm going to try to turn that into a strength by hopefully getting everybody to do different things and look at a different set of symbionts and maybe collectively gain a sense of the diversity of what's out there. So, as I've said, I have not printed a handout as yet, but one will go out this afternoon. And the first thing that you're going to have to do is get a bug. There are lots of ways you can do this. I mean, this is Arkansas, we are not short of bugs. We've got, plenty of bugs out here, nothing to share. I also don't necessarily want you going outdoors more than you have to, but if you have a place where you could catch crickets or grasshoppers or something like that, that's great. I was just coming to that. If you have occasion to go to a pet store, don't make a special trip or expose yourself to unnecessary risks or spend money that you can't that you can't spare. But you could use feeder crickets, or mealworms work well. Or for those in Conway, the Petco, the new one, close to Michael's and Academy Sports and all that. They've got several varieties of feeder cockroach, if you wanted to go that route. If you're close to a place that sells live bait, bait crickets would work just fine. If you want to go ahead and catch your own, crickets, grasshoppers, roaches would work fine. Flies potentially could be interesting. They can be a little tricky to catch, but you can make a homemade bug trap just by taking a plastic soda bottle, whatever size, doesn't really matter, cutting the top off, turning the top upside down, and placing it back into the body of the bottle. So you have a bottle with this cone pointing inward, and bugs can enter that, and then they can't figure out how to leave. And you can tape the seam if you need to, or you could just use paper clips or something like that to hold it all together, or you could just push it in firmly enough that ordinary pressure will hold. But that's a nice way to make a quick and dirty insect trap. And then, of course, you put bait inside. So I've caught fruit flies before, just with some old bananas or something like that. You can catch other flies if you put a bit of meat down in the bottom, especially if it's meat that's getting, kind of whiffy. That hamburger that you bought 2 weeks ago and never quite got around to using, that sort of thing. You could try other kinds of trash as well. See if you can catch house flies or some such. So you can make simple traps like that. You can try catching them by hand. One thing we did in parasitology this past spring that worked really well was I brought in a nice bucket full of pond water that I'd kind of scraped up off the bottom of, it was actually Lake Beaver Fork. I'd actually kind of scraped the bottom and brought up some mud and some vegetation, and that turned out to be rich in insect larvae. In particular, we had, now I can't remember what they are, some insect larvae that had what looked like 3 feathers sticking out of their butts. And I can't remember what group of insects those belong to. Bad biologist, no cookie. But they turned out to have interesting things in their guts. And what I did this morning was just go pick up some rotten wood And backlit like this, I don't know if you can see it, but it's got lots of tunnels with termites in it. And termites have really interesting critters living in their guts that actually aren't parasites. They're they're mutualists. The termite needs them for proper health. Okay, so once you've got your bug, if you're squeamish about squishing the intestine out of a living insect, or if the insect is flying or scurrying and you're having a hard time handling it, simplest thing to do is put it in the freezer for about 30 minutes. That's not long enough to freeze it solid. We don't want that, but it will knock them senseless, although it only lasts for a few minutes. Once you take them out of the freezer, you're going to have to work fairly quickly because they will start coming back to life. But that will knock them senseless long enough for you to, cut the head off and remove the intestine without too much hassle. If you've got a big insect, you may use your dissecting tools. For a big roach, probably the easiest thing to do is just cut up the sides of the abdomen and then just kind of open it up like a book and tease the intestines out with a probe. If you've got a smaller insect, the easiest thing to do is probably going to be just to take your tweezers and maybe cut off the head or pin the head down with your probe and apply gentle traction on the butt with the tweezers. And if you do it just right, the butt end and the hindgut will come out in one piece. You'll get the butt end coming out, and there'll be this little gut kind of trailing along behind it. That works really well with mealworms. It's probably what I would try with feeder crickets that Wendy says she's got and that some of you might want to try. It seemed to work pretty well with those aquatic insects that we had. It may take a couple of tries to get right, but it ends up working pretty well. So you want to get that gut out of the insect. What I'm going to try doing is, for a really small insect, the easiest thing might be to squish the whole thing. With termites, you can be just have a little bit more finesse. You don't want to look too much at the intestine itself. We want to look at what's in it. So you can take a termite, and if you can hold it down, you can apply very gentle pressure with the probe, not the pointy part, just kind of rolling the side of the probe on the body and squeeze the gut contents out. And I'm going to see if I can do this. So there's a slide, and let me get see what tools I've got. Yeah, there's a probe. There's forceps. And let me see if I can. Okay, I've got a roly-poly on this wood. Right now I'm just kind of breaking down this decaying wood and seeing if I can get Mr. Termite to pop out. A roly-poly is big enough that I would personally probably want to put him in the fridge for 30 minutes, but termites you can do fairly quickly. Here, termy. All right, I'm gonna knock some of these guys back into the bucket so they don't crawl around and freak out the cat, who is right next to me, of course. Ah, that one. What's this? Okay, this is not who I was expecting, but this is... I can't really see it clearly because I think I just mashed up. No, you don't get this. This is not kitty food. Yeah, I can't quite tell what this guy is. I think this might be a wood roach nymph. So I could try that trick of holding him down and pulling his abdomen, and I think that's what I'm going to try. So we'll actually... Hi, cat. Hey. Cat, please get off the table. Good kitty. Okay, so I'm going to kind of put a probe through the thorax and try to pull the abdomen and there. I've just got all of the guts to come out in one piece. So that actually worked fairly well. Now, what we need to do is add some fluid, and if we were in regular lab, I would have saline. And the cat just stepped on the slide. Animals are such a blessing in our home. Maybe some of you are in the same boat. Yeah, Wendy, I envy you. Your fish don't mess with things on the counter. Fortunately, the gut's still there. So we want to add a little saline. And the saline that we'd ideally have would have a number of salts in it, magnesium and potassium, as well as sodium and chlorine. What hopefully will do for our purposes is a 1/4 teaspoon of table salt in 1 cup of water. But I have to cut the salt in half because the cat drank half the water. So I have about 1/2 cup of water. So I'm putting in about 1/8 of a teaspoon of salt, and I'm going to put in the tiniest pinch of baking soda. That should do. And it's not going to work as well as the stuff we'd normally use in the lab that has glucose and magnesium chloride and things like that. It's not going to work as well as insect ringer solution would, but it'll do. And by the way, you should probably mix this up before you squish your bug. I should have done that. Bad professor, no cookie. So we squish that up, and I'm going to put 1 drop on this gut. And I'm going to put a cover slip on top. Actually, I'm going to do one thing first. I don't just want to look at the gut. I want to look at what's inside it. So to get the gut contents out, if you've got a large gut, you can try kind of milking it. In fact, I think that's what I'm going to do here, given how small this thing is. I'm going to put the cover slip on, but I'm going to use the point of my probe to press down on it and maybe see if I can't strip it a little bit. With mealworm gut, you can kind of milk those just to try to squeeze out as much gut fluid as possible. or you can kind of use you can use the blunt end of the probe and just kind of mash down a little bit on the gut like that, or kind of strip it like that. Shivam and Khazer, you've been in a class where we did this a couple of times. Do you have any pointers on technique? What's the best way to squeeze out gut contents?
Just do your best.
Okay, and be prepared to do it more than once.
Oh yes, definitely. I think
Okay.
that's what I was thinking too, having more than one bug actually.
Yeah, it may take a little practice. And again, I'm actually curious because I've done this before where the entire class was just looking at one species. I'll usually use mealworms for this, or I'll use South American orange-spotted roaches, because there's a very nice lady who lives up in Quitman who farms those because people buy them as food for pet lizards and things like that. Or sometimes Dave DeSword has feeder crickets I can use, but yeah, we'll usually end up with everybody looking at the same critter. But it would be interesting, I think, to compare notes and just look at, symbiont diversity in insects around here. So some of you can do feeder crickets or things like that, but we don't all have to. Okay, so I've got this on the slide, and I'm looking right here at the squished up guts. So there we are. And I'm actually going to go out of focus just a little bit from your point of view, because what I can see, the focus of the scope with my bare eyes is different from the focal point of the camera. So we want to see if we can spot anything moving. Okay, the stringy stuff right there that looks like spaghetti, that's actually part of the bug. That's called the Malpighian tubules, and they are hollow excretory organelles that branch off of the junction between the midgut and the hindgut, if memory serves. So you'll see parts of the insect. You may also see things that look kind of like annulated tubes, a little bit like dryer hose or maybe vacuum cleaner hose or something like that. That's probably spiracles, tracheas, the breathing tubes of the insect. You might see blood. You'll certainly see gut contents. So some of what we're looking at is the remains of this unfortunate bug's last meal. Some of it right there. Yeah, this kind of hard brown stuff that we're looking at is remains of the insect's cuticle. That's the remains of the butt where I pulled on it with the tweezers. So we're looking around. What can be found? Doodly doodly.
And do you want us to be looking at something particular in the underneath the slides?
What do you mean for something in particular?
Like, what are we supposed to be looking for exactly?
Okay, what I will do is in the handout, I'll send you pictures of everything I've got of the things that live inside bug guts. I was hoping I'd have some for you right now. One very common thing that people find is gregarines. If you remember, we talked about those briefly yesterday. Okay, there's lots of little bacteria and things in here. Gregorines are those apicomplexans. They're cousins to malaria and things like that. Gregorines are that looks like a head end and what looks like a body end. And those are fairly common. And if you didn't catch them in the lecture, as I said, I will send out some pictures. All right, I'm going to go out of focus a little bit again to try to zoom in on something cool. If you get termites, you may also, you'll find a lot of flagellates swimming around. Termites have really, really cool gut biotas of symbionts that actually do the work of breaking the wood down. We'll actually talk about those guys in lecture tomorrow. And I'll send you pictures of what those look like as well. In the roaches, there's actually some large ciliates called Nyctotheris. And I'm not seeing okay, I'm not seeing much here. One thing I'm seeing that you're not looking for is if you get an insect gut out, it very often has a lot of fat stuck on it. And so anything that looks kind of yellow and greasy, you probably ought to try to scrape off if you can. And these little spheres right there, those are just bubbles of fat. Insects store their fat in these things called fat bodies. And if you get any of that in, you'll see lots of that sort of stuff. I'm going to go to low power again for just a second. Okay, so not an awful lot seems to be visible in this particular insect. We have fat and gut contents and remains of the actual gut. But I'm not seeing a lot in here other than squish bug guts. So I'm going to do one more and see if I can get an actual termite out of here. Hi, Fern. I tell you what, if you guys would like to take about a 10-minute break while I try to grab a termite and see if I can squish it out, you've seen basically what technique there is, and there really isn't any one fixed technique, and it gets a little bit messy. But if you guys want to take a 10-minute break, I'll grab a termite out of this wood and squish out its gut contents, and we'll see what we can see. So I will I've got 9:38, so at 9:50, hopefully I'll come back. For now, I will turn off the mic and the camera, and you guys go get yourself some caffeine.